Introduction to Bioinformatics, Part 2. In this part we're going to look at sequence alignment, homology searching and annotations. What do I mean by sequence alignment? Well, all of these three examples are ideas of what we could mean by sequence alignment. So in this example we're aligning the C-terminal end of these sequences, LD, S and L. In this example, we're aligning the N-terminal region, G, S and L. Or we could even mean this, where we're aligning the S and L at the end of this and with the beginning of this. But generally what we mean when we talk about sequence alignment is introducing gaps into one or both sequences in order to optimize the number of matches that we have. So in this example we could add a gap here such that the L in the second sequence aligns with the first L in the first sequence. Or we could introduce it here such that the L in the second sequence aligns with that second L in the first sequence. Now, given just that information, we do not know which of these is better or correct. Both of them are equally valid. If we had multiple sequences, we might get some clues as to whether one of them is more correct than the other, and indeed if we had a structure, we could see structurally where that insertion or deletion occurs. So how do we actually analyze sequences in order to get this information? Well, the key concept is what is known as a dot plot. And all I've done here is to take the first sequence and plot it along the top, and the second sequence and plot it down the side. And everywhere where there's an amino acid match, I've put a red blob. So what you should be able to see if you sit back a little bit from the screen is that there is an approximate diagonal with something weird going on in the middle here. And this is where the insertion in the top sequence occurs with respect to the side sequence. So the process of aligning two sequences automatically essentially starts with doing this and then automatically finding the best path through the matrix in order to align as many residues as possible. Now this is an example of a real dot plot, and here I've taken two light chain sequences from antibodies. So we're looking at a region of around about 110 amino acids, and one is plotted along the top, one down the side, and you can see in this left-hand sequence an approximate diagonal, but it's quite noisy and difficult to follow. What I've done then on the right is a method of noise reduction where I've only plotted a point if there are 3 out of 7 residues that match. And here now you can see a much clearer diagonal going down the middle here. Those dot plots were based on sequence identity, and as I mentioned in the first half of the lecture, looking at sequence similarity can often be a better thing to do. So in a simple identity scoring, we score 1 for a match, 0 for a mismatch. In these more complex scoring systems, we account for the similarity between amino acids. For example, serine and threonine are very, very similar to one another. The only difference is that threonine has a little CH3 group stuck on the side. That means that they're generally able to substitute for one another during evolution, whereas, for example, serine and tryptophan are very different amino acids and much less likely to substitute. So typically these similarity scores are derived from an analysis of aligned homologs and looking to see what substitutions are observed. This was first done by Margaret Dayhoff in 1978 and improved by Henikoff and Henikoff in 1992 to produce the so-called Blossom matrices that are generally used for doing searching of protein sequences against large databanks. Now, this is the Dayhoff matrix, and just to look at some examples here, we're plotting the one-letter amino acid codes along the top and along the side, and then the score in the matrix. So this is a log odds score. That means observed over expected, and then take the log of that. So we get negative scores for things that are less common substitutions than you expect by chance, positive scores for ones that are more common than you expect by chance, and 0 means that you are seeing that substitution at the rate you would expect purely by chance. So let's look at a couple of examples. D and E, that's aspartic acid and glutamic acid. So asp substitutes for itself, in other words it's conserved, much more frequently than we expect by chance. We get a score of 4. A replacement with glutamate, we see a score of 3. So still, we see that much more frequently than we'd expect purely by chance. If we compare glutamate with, say, glycine, we see a value of 0. If we take cysteine versus cysteine, because remember that cysteine can form disulfide bonds, we see a value of 12. So cysteines tend to be very, very highly conserved. Similarly, tryptophan, we see a very high positive score. Tryptophan, remember, is very much larger side chain than others, so that tends to be very conserved. And interestingly, one of the lowest scores is for cysteine to tryptophan, score of -8, indicating that these very conserved cysteines are virtually never replaced by very conserved tryptophans, and vice versa. As I mentioned earlier, the process of doing automatic sequence alignment involves looking at one of these dot plots and tracing through it to find the optimum path. And the method that's used to do that is known as dynamic programming. So what we do is apply this dynamic programming algorithm to generally a similarity scored dot plot, find the optimum path through the matrix, which maximizes the scores that we get while minimizing the number of insertions and deletions in order to find the best alignment between the sequences. Now, this was introduced initially by Needleman and Wunsch in 1970. And Needleman and Wunsch were two clinicians. They came up with this idea. They published this seminal paper in Journal of Molecular Biology in 1970, which is probably one of the most widely cited papers in bioinformatics. And then they kind of disappeared back into clinical medicine and never published anything again. Smith and Waterman then realized that what Needleman and Wunsch had done was dynamic programming, and they formalized what had been done previously, because Needleman and Wunsch had actually used a type of alignment called global sequence alignment, and when Smith and Waterman looked at this, they introduced a variation on the algorithm that produces a local sequence alignment. Now, a global alignment essentially takes the whole of one sequence and tries to align it against the whole of another sequence. Whereas a local alignment takes a subregion of one sequence and aligns it against a subregion of another sequence. And it does that completely automatically. Now, why is that useful? Well, it's all about the way that inheritance works. And we can think of domains in proteins as being units of inheritance. So this is showing on the right a set of different protein sequences, but the domains are coloured based on conservation. So all of these sequences contain one or more of these red domains, which are adenine nucleotide alpha domains. And you can see that these are paired then with other domains. Now, that means that it would make absolutely no sense to try to align this sequence where we have a yellow domain followed by red domains with this one where we have a cyan domain followed by red domain. So thinking of a domain as a unit of inheritance like this, if we have domain A associated with domain B in one protein and domain A associated with domain C in another protein, if we tried to do a global sequence alignment between those, then okay, A would align with A, but B would be aligned with C. And that makes no sense at all because domains B and C are not homologous to one another. So if one did a local sequence alignment, the algorithm would realise automatically that A aligns with A and we can ignore the alignment of B with C. So let's take an extreme example of that where we have domain A followed by domain B and we're wanting to align it with domain B followed by domain A. Now, a global alignment would try to do just that. It would align A with B and B with A, which makes no sense whatsoever. Whereas a local sequence alignment would align A with A or B with B, depending on the parameters and the order in which you put the sequences into the alignment program. Let's have a look at a real example of that. So rather than a domain, I've taken a short stretch of sequence. So I've got this sequence starting ASD, which is our domain A, and this one starting DPR, which is our domain B. So in sequence 2, I've taken our domain B followed by domain A. And these are actually identical sequences. So ASDH exactly matches ASDH here. If I try to do a global sequence alignment of those, then I end up with something which actually doesn't look too bad, but of course we know is meaningless because these are not related. A is not related to B and B is not related to A. So if we cut off the tails, in other words the bits at the ends of the sequences which don't align with anything, then we have 48.4% sequence identity. Now if I do a local alignment, then I would get something like this, where again if we cut off the tails we have 100% sequence identity. Now just to talk about what we mean when we score sequence identity, it depends really what you divide by. Now sequence similarity is calculated by accumulating those scores across the matrix when we're doing the sequence alignment and then dividing that by the lower of scoring sequence A against itself and sequence B against itself, because you can't score higher than that. When it comes to calculating sequence identity, the question of what do we divide by arises. So one option is to calculate the number of matches, in this case 15, and divide by the overall length of the alignment, 34. Another option, which is what I showed you in the previous slides, is instead to divide by the alignment with no tails. In this case that would be 15 over 31. Frequently what most people tend to mean if they just say a percentage sequence identity is the identity over the shorter sequence, because as with the percentage similarity you could never score more matches than the length of the shorter sequence. In this case the two sequences are the same length, they're both 28 residues long, so it doesn't matter which sequence we choose, and the score would then be 15 over 28. Another option is to score over the aligned residues. So in this case we've got 22 positions which are aligned with one another, 15 of those matched, so the score would be 68.18%. The other thing that's quite often done is to score the alignment over one chosen sequence. So in this case, I'm scoring over sequence 1. And that can be important when you're doing things like homology modelling. You've got a target that you wish to model, and you want to know what percentage of the residues in that sequence are matched in the template that you're using to model it from. A couple of other statistics that we can calculate: the number of deletions in sequence 1, and that's, in this case, 6 deletions occurring at 5 sites. So 1, 2, 3, 4, 5. And then we can also calculate the insertions in sequence 1, which would be the same as the deletions in sequence 2. So in this case, 1, 2, 3, 4, 5, 6, occurring at 4 sites, here, 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 and here. Now, this concept of aligning sequences can be extended to looking at multiple sequences. And that's useful in a number of ways. I mentioned previously where we had that ambiguity about where an insertion or deletion should be, that having multiple sequences in an alignment could help us to see where that gap actually should occur. But also, having a multiple sequence alignment allows us to find conserved sites. So in this alignment, those are indicated with a star, where we've got 100% conservation in this set of sequences. And we have colons where the sequences, the residues in the sequences are all very similar, even if there's some divergence, and dots in cases where we've still got some similarity, but it's reduced. Now, once we have something like that, we can also do the calculation of phylogenetic trees. We've talked a lot about homologs, so how can we identify them? Well, what we want to do is have a sequence of interest and then be able to search a data bank of some sort to find related sequences. Now, as I mentioned earlier, although we generally say in every case if we have at least 35% sequence identity over at least 100 amino acids, we can be sure things are homologs, we also see homologs with much lower sequence identity, perhaps 10% or less. But random sequence pairs can show up to 20% sequence identity. And these can be cases where we know that they are not homologs because, for example, one's an all-alpha protein, one's an all-beta protein, and they have completely and utterly different functions. So above around 40% or so, automatic pairwise alignment in general is enough. If the sequences are long enough, we can go to somewhat lower sequence identities. In that kind of lower area of around 25 to 40% sequence identity, we can use so-called consensus methods. And this is really looking for consensus conserved patterns within sequences. Below that, in the so-called twilight zone, we can use profile methods. And this is an extension of the consensus method where we're looking at the frequency with which each amino acid occurs at a given position, rather than looking at the expected, most likely amino acid at each position, which is what we do with the consensus method. And at the lowest sequence identities, we may be able to recognize homology by predicting the three-dimensional structure and also looking at the fact that we have some functional similarity. Now, going back to the basic pairwise sequence comparison methods, the problem with searching these huge data banks that we now have is that dynamic programming is slow. And therefore we use special methods like FASTA and BLAST, which are fast approximate methods known as heuristics. So a heuristic is not guaranteed to get the very best match, but it is guaranteed to get a very good match. What we do in both FastA and BLAST, the details of the implementation are quite different, but the concept is very, very similar. What we do is index the database by finding locations of short words in the sequence. We then take our probe sequence that we're searching with, and we find those words in that probe sequence. We look for multiple matches and extend those to find likely hits in the databank, and we can then do a Needleman and Wunsch or Smith-Waterman dynamic programming alignment on just those likely hits. So you can think of this like having a book, and the book has an index, but instead of indexing just important words or phrases, it indexes every word that's in the book. So if you wanted to find a particular sentence and what page it occurred on, you would simply look at each of the words in your sentence, find which pages those occur on, and then find the pages that have the most matches. What you could then do is go to the pages with the most matches, read those pages in order to find your sentence. Now, it wouldn't matter if the sentence that you were searching for wasn't exactly correct. If you had some mismatches, you'd still be able to find the right page and recognize your sentence. So the finding the pages part is the heuristic method, and then you use the dynamic programming to do the searching of the pages to find the exact match to your sequence. Now, there are important statistics related to this. So the number that is normally quoted is known as the E value or expectation value. So let's take a normal die. What is the P value, the probability for throwing a die and getting a score of 6? Well, obviously it's 1 in 6. What then is the p-value for throwing a 5 or more? Well, there are two ways you could do that. You could throw a 5 or you could throw a 6. So it's 2 in 6 or 1 in 3. Now, if you were to throw the die 12 times, how many times do you expect to see a score of 5 or more? Well, that's the expectation value. It's calculated simply as the probability, in this case 1 in 3, multiplied by the number of times you throw the die. So that would be 12 divided by 3, which is 4. So if you were to throw a die 12 times, you would expect, purely by chance, to see a 5 or 6 4 times. So we can define then the expectation value, the E value, as the number of times you expect to see a hit with a score of X or better purely by chance in this database. So if you're doing a search with a sequence against a resource of other sequences, you can calculate for each score that you get what's the probability that you see this score or better purely by chance, and then you multiply that by the number of entries in the database, and that gives you an E value. So you can calculate a P value and you can calculate an E value, but it's normally the E value that is cited because that is more useful. It's referring to the number of times you expect to see this hit in this database purely by chance. Consequently, when you see really close homologs with very high sequence identities, you will typically see E values of 10 to the minus 100 or smaller. But in general, we take a threshold of something like 0.1 and say that if we have an e-value of 0.1 or less, then there's a good chance that this is actually a homologue. Now, of course, as the sequence databanks get larger and the searches that we're doing are then on these larger resources, the n in this equation gets larger and consequently the e-value gets larger. So when we do a search 1 year and get an e-value of 0.1, we do that search a year later, we might have an e-value of 0.2. However, as I mentioned, at low sequence identities we can still have homologues. And we can generally identify these because we have functional residues that are conserved in a kind of sea of variability around them. So looking at this sequence alignment, it's quite hard to see that there's any relationship here. But if we seek out where the conserved residues are, you can see that there are positions here that are actually highly conserved. And that's what profiles allow us to do. They allow us to identify the frequency with which each amino acid is seen at each position in an alignment. And that profile can then be used to compare against another sequence to find out if it matches. As I mentioned earlier, the kind of simplification really of a profile is to use a consensus pattern. And that's what some of the secondary databanks such as ProSite do. Now, ProSite contains two types of consensus pattern. The first type is for functional sites, which can be useful in terms of function prediction, and the second type is for homologous family characteristics. So this first example is what in computing we call a regular expression. So the square brackets mean or. So this means a serine or a threonine followed by any residue followed by arginine or lysine is a pattern that is characteristic of a protein kinase C phosphorylation site. Now the curly brackets mean not. So this site is asparagine followed by not proline, serine or threonine followed by not proline. And that's characteristic of an N-linked glycosylation site. Now in both cases it doesn't necessarily mean that just because you have that sequence pattern that these will be those sites. They would have to be, for example, on the surface. But every N-linked glycosylation site and every protein kinase C phosphorylation site will have these patterns. Now the example here for a homologous family characteristic is for a particular domain called a Kringle domain, and here we have a slightly more complicated pattern, but we're seeing either phenylalanine or tyrosine followed by cysteine, followed by arginine or histidine, followed by asparagine or serine, and then the round brackets give you a number range. So this means between 7 and 8 of anything, followed by a tryptophan or tyrosine, followed by a cysteine. So every Kringle domain has this pattern. Once again then, we can use these types of homologous family characteristic patterns in order to look at a new sequence and identify whether it might be a member of a particular family. And in this case, we might be looking at sequences which overall have very, very low sequence identity to other members of the family, but they have these conserved patterns. Finally, we'll go on to annotation, which is the human factor in all this. What do I mean by annotation? Well, things like references, citations, methods used, cross-links between different databases and databanks, so-called feature tables, which indicate segments of biological significance, such as for DNA, coding regions, for proteins, might be enzyme active sites, and so on. But also more general descriptions. And the problem with these are whether or not they're going to be computer parsable. So if we have a general description, it's easy for you to sit down and read as a biologist, but in order to analyse across different sequences and so on, it's quite difficult for a computer just to look at a block of text and look for features in that text. So in some cases these annotations may use so-called restricted dictionaries, where we guarantee that we only use a limited number of words coming from this set that we have defined. And an extension of that is a so-called ontology, which is technically called a directed acyclic graph, and this means that it is a bit like a tree, but a tree where things can have multiple parents. So in a normal tree we can have things that inherit the characteristics of a single parent. For example, we might have animals. Mammals then have their own characteristics, but also have all the characteristics of their parent, which is animals. Then we might have mice, which inherit all the characteristics of a mammal, plus particular characteristics for a mouse. We might also have a giraffe that would inherit the characteristics of a mammal, but not those of a mouse. Now in an ontology it's exactly the same. We have words which inherit the characteristics of their parent word, but also can inherit from multiple parents. And in annotation we also mean things like the authors, the people who've sequenced a particular gene or protein. Now, the quality of the raw data that we have in a primary data bank is as good as the methods that produce it. Now, for purposes of putting sequences into these sorts of resources, peptide sequencing has largely been replaced by a DNA sequencing, which is now fully automated and therefore much better and more reliable than sequencing information was some years ago. However, the quality of the annotations is only as good as the curators and annotators who write those annotations. So in the pre-genome world, if anybody actually bothered to sequence some DNA or a protein, then that information came from a single research group who had a particular interest in that gene or protein. Consequently, the annotations were grounded in experiment and written by experts in that particular gene or protein. In the genome world now, however, there is rarely experimental confirmation of the expression of putative genes or the characterization of their products. Instead, annotation can often be based on computer analyses, and this has been described as the weakest link of genomics. Getting it right is labour-intensive, underfunded, and scientifically relatively boring. But people have said that poor annotation really negates the benefit of having genome sequence data at all. Here are a few quotations. So Michael Ashburner, who was at the European Bioinformatics Institute and led the Gene Ontology Consortium, which created these ontologies for describing proteins, said, "GenBank is full of mistakes." Douglas Crawford from the University of Missouri, "The utility of GenBank has declined greatly." And Terry Atwood, who's at Manchester and author of the Prince secondary data bank, and it used to be at UCL, said, "We have to educate people about databases so that researchers don't assume that they are right." Now, just a few examples. This quotation from Keith Yamamoto: "Scientists would rather share each other's underwear than use each other's nomenclature." So this is an entry from Swissprot, where this particular entry, TUP1 yeast, has 10 different names for the gene, because different groups have identified it and given it their own name. Even worse, here are two different proteins, MRF1 yeast and RF1M yeast, but they have been given the same gene name. So obviously this can cause all sorts of problems. Now, as a result of that, as genomic data grow and methods improve, resources sort out these sorts of problems, then obviously the annotations will improve. However, the corollary of that is that annotations will be in a state of constant change. And does that mean that research projects that have relied on earlier versions of data may have to be revisited and checked? Now, Arthur Lesk, in his 2002 book on bioinformatics, and there have been several editions of that since then, said, "Databanks will become a seething broth of information, growing in size and maturing, we must hope, in quality." So at that point I'm going to finish and summarise with the key take-home messages that you need to know from this lecture. First of all, the difference between databanks and databases, the difference between a primary databank and a secondary databank, some examples of both, the definitions of homology, orthology and paralogy, and why homology in particular is important, the difference between a databank like Swissprot and a search tool like BLAST. So when you're searching something like Swissprot, because it's a databank, you can choose which tool you use to search it. So you might choose BLAST, you might choose FASTA, you might choose some simple text search. The general concepts of local and global sequence alignment and why local sequence alignment is important, the basic ideas of sequence searching and the meaning of the E-value, and finally some of the problems of annotation.